Hello students, welcome to the academia. So in our last video I have discussed about vitamin A and the biochemistry, the structure, and about retinol and retinoic acid, the absorption, transport and mobilization process. So in today's video there is the biochemical functions of vitamin A. So vitamin A is necessary for variety of functions like vision, which is most important, then proper growth, and differentiation, reproduction, and maintenance of epithelial cells. Okay, so these are the most important functions of vitamin A. In recent years, each form of vitamin A has been assigned specific functions. Okay, so first of all, if we go through the vision, generally all of you know that for the eyesight, from the very childhood we are advised to having a vitamin A or the foods rich in vitamin A. Okay, so what is the biochemical process? So the biochemical function of vitamin A in the process of vision was first elucidated by George Wald. So the name of the scientist was George Wald, and he was nominated for Nobel Prize in 1968, having Nobel Prize 1968. Okay, and the events occur in a cyclical process known as Rhodopsin cycle, or it is also known as, according to the name of the scientist, that is Wald's visual cycle. Okay, so basically rod cells and cone cells, what are these? So if we go on through the mechanism, so rods and cones are what? The retina of the eye possesses two types of cells. So basically rod and cone cells are present in the eye retina. And the human eye has about 10 millions of rods and about 5 millions of cones. The rods are in the periphery while cones are at the center of retina. So when you go through the structure of retina, when I will discuss about the retina, then we understand the structure of retina, the physiological feature of retina, that the rod cells are basically present in the periphery while the cones are at the center. Rods are involved in dim light vision. So what is the function of rod? That is, they are involved in dim light vision. Okay, and the cones are responsible for bright light and color vision. And color vision. Okay, so generally, so this is about humans. So animals like many of you know that owls and cats, for which night vision is more important, possess mostly rods. So in case of a human eye, so this is the eye retina of human, having 10 billions of rods and about 5 millions of cones. So this is about, and it is responsible for dim light vision and it is responsible for bright light as well as color vision. Now what about the Wald's visual cycle. Okay. So now see the cycle. So this is the Wald's visual cycle. Rhodopsin. Okay. Rhodopsin having the molecular weight of 35,000. Okay, and it is actually a conjugated protein present in rods. So where from the rhodopsin come? Actually the rhodopsin come from rod. So from the rod the rhodopsin come, and rhodopsin basically a conjugated protein. It contains, and it is present in the rod cells. It contains 11 cis retinal and the protein opsin. So the rhodopsin contain 11 cis retinal and opsin. Okay, so these two components. The aldehyde group is linked to the amino group of the lysine of the opsin. Okay, so the retinal having the aldehyde group. So if we go through the chemical structure, I have not showing here the chemical structure. If I go through the chemical structure, so it having the aldehyde group. Okay, the aldehyde group, and the opsin having the amino group of lysine, and the opsin having the amino group of the amino acid lysine. So both of them make a compound that is 11 cis retinal opsin. Okay. The primary event in visual cycle. So just to see the step-by-step events. The primary event on exposure to light. So first of all, if the light exposure is there, that also we call it photon, is the isomerization of 11 cis retinal. So this isomerized and to all trans retinal. So the 11 cis retinal by the enzyme retinal isomerase, it form all trans retinal. This leads to a conformational change in opsin. Okay, so this kind of conversion actually change the conformational change in opsin, which is responsible for the generation of nerve impulse. So here we get the nerve impulse. So light, which is the stimulant, and then this kind of reaction occur and we get the nerve impulse. The all trans retinal is immediately isomerized by retinal isomerase to 11 cis retinal. Okay, so you can see the combination, this combines with opsin to regenerate rhodopsin. So can see the thing here. So all trans retinal, it is actually immediately isomerized by retinal isomerase, and again we get the 11 cis retinal. This combines with opsin to regenerate rhodopsin and complete the visual cycle. Okay. So this is actually what happens here. However, the conversion of all trans retinal to 11 cis retinal is incomplete. Therefore, most of the all trans retinal is transported to the liver. So this portion actually, it is, you can see this portion, so which occur in the liver, and converted to all trans retinol. So this conversion is done by alcohol dehydrogenase. So this, when it goes to liver, by the liver enzyme, there is formation of all trans retinol, from all trans retinal to all trans retinol in liver by the liver enzyme. the all-trans retinol undergoes isomerization. See, it is also by the enzyme present in liver, isomerase to 11-cis retinol, which is then oxidized to 11-cis retinal to participate in the visual cycle again. Okay, understand? So that was basically the Wald visual cycle, but when this goes to the liver, by the liver enzyme there is conversion from this to that, and then it is again by the liver enzyme isomerase converted to 11-cis retinol, and this 11-cis retinol again converted to cis retinal, and by the enzyme alcohol dehydrogenase, and again it participates in the visual cycle. Okay, so this is the thing, or this is the cycle actually. So regarding the function of vitamin A, regarding the vision or visual function, this rhodopsin cycle or the Wald visual cycle is the most important, and this is the mechanism how the cycle actually takes place. And in our next videos I will go through the other functions of vitamin A, as I have already discussed, that proper growth, differentiation, reproduction, maintenance of epithelial cells, etc., etc. And also will focus on the color vision by the cone cells. So hope you people can understand. If you have any query or any question, you can ask or can comment in the comment box. Thank you so much.